Okay, thank you. And so I'm also excited to being here, and I'm also excited to learn, and I hope we'll have a good discussion at the end. If there are clarifications while we are going through, please just put them in the Q&A box, and Missy will then speak out, and then we will have some of the hopefully longer and more practical discussions at the end. So please add all kinds of questions and comments and suggestions and what you're thinking about into the Q&A box so we can either discuss today or follow up afterwards. So as I said, Suya Kao is the graduate student who's been doing a lot of the work that we're seeing here. A lot of you might know Alan Davis, my good colleague, 2 doors down the hallway, who has been doing a lot of stormwater work with nutrient removal the many decades. And then you can see my email there also if you would like to be in touch. So what we'll present today is the results that we have from the last couple of years where we have done work sponsored by the Pool Monitoring Project of the Chesapeake Bay Trust, where we're focused on land use and area developments to understand some of the pathways of where the PCBs that we end up seeing in the environment and eventually in the Chesapeake Bay, where they're coming from and what our options are to deal with them before they get out into, hopefully not the environment, but get to source, treating the sources earlier on. So for just a short recap on what are we dealing with here? Well, we're dealing with polychlorinated biphenyls, and there is a reference in old literature that the first trace of these were found back in the 1800s in coal tar in the Appalachian Mountains in coal mining. At that point, it was traces, and it shows that it's a natural group of contaminants. They're just or group of compounds that have now become contaminants because they've been mass produced up until the 1970s, where they were banned from production as the main product in the United States. It falls together with the development of the EPA and a lot of our Water Act and so on in the 1970s. There is already there an acknowledgement that there isn't direct impact on the nation's waters. And what we're talking about are a group of contaminants where the chemical structure that you can see here is made up by 2 benzene rings where there are a number of chlorine atoms that are added on. And depending on how many we have of the chlorines, then we can have up to 209 congeners that can be mixed in different ways, as we'll see a little bit later. They can twist around that chemical bond in the middle, which makes we'll talk about in a little bit, they can impact the food chain in different ways and the environment. They have been used in very many commercial and industrial applications throughout their prime time from around 1920s when what became Monsanto started mass producing these. And as you can see here, just a little exhibition of where we know that they have been used. On the upper left side, you see the gravel road. Early on, before it was known how toxic they were, it was used as dedusting agents because these compounds are hydrophobic. So they would bind to the dust particles and they will settle much faster. So there wouldn't be the cloud of dust if, for instance, a car or other would drive down the gravel road. And that is the exact same mechanism that we see in water, that the PCBs bind to particles because of that chemical characteristic. The other, from a chemical point of view, great characteristic is that these do not burn easily. So they're thermostable, which is what they're being used or have been used for in transformers and in industrial equipment, as you can see on the right-hand side. And it's a fairly old estimate, but there is none that's newer. That is between 0.6 and 1.2 billion kilos of PCBs that are out in the environment. And as illustrated here, it's also made it to the Arctic areas, even though the PCBs were not really used in those areas, but water and air and human activity have made PCBs move all over the planet. So why do we care about this? We care because, as you can see at this figure at the beginning, they bioaccumulate and biomagnify in the food chain. And it's a similar story to the silent spring with DDT in the 60s, that the slightly different mechanism though, but they end up and impact bald eagles at the top of the food chain. But we happen to be there too. And a lot of this is very important for us in the Chesapeake Bay area because many of these steps in the food chain are important for not only recreational purposes, but also the economy of the state. Eventually, PCBs will end up in sediments and soils, as I said, because they're hydrophobic. So we know where to find them. And we'll see in a little bit that we do indeed find them there a lot. Canada has been heading up the human research in this and have found that there are basically none of us where PCBs cannot be measured in either our tissue or our blood because of the many products that PCBs have been used in. And the piece of this that's really bad, of course, is that they're toxic. They have, depending on the different kind of congeners that we're dealing with, they have different kind of toxicological effects. So they can induce cancer. There can be issues with the endocrine system, reproductive organs. There could be immunological issues, and there could be mixes of these. And one of the reasons is that the chemical structure of the compounds can mimic certain hormones, and then the PCBs take up the space of what that hormone should be doing in the cell, and then we of course get a different response in our body. We are not the only state or the only region where PCBs are the main cause of water impairment. As you can see here on the grainy image, this is also a fairly dated map, but unfortunately it's still the message is still up to date. And you can see that on the East Coast, meaning Maryland, Virginia, our area, PCBs are the main cause of impairment, at least here in lakes and reservoirs and ponds, but it's the same for creeks and rivers. Then you can see further down south, there are a couple of places, Alabama and Georgia, where their PCBs are issues which relate to the early production of the PCB and the manufacturing companies that were placed there. And then if we go to the left side, we see Washington State, where PCBs are also a really big deal. And a lot of the knowledge early knowledge comes from Washington State because they have been very early out, because the Puget Sound and the fisheries there have been impacted a lot by PCBs. There is a big naval activity around the Puget Sound and Seattle, Tacoma, which is also where a lot of their PCBs are coming from. The map does not say that because we do not see PCBs listed in other places that they are not a problem there. The map just shows that PCBs are not the main problem or the main cause of impairment in these other issues, because we do know that in more or less every state, there are Superfund sites, there are historical sites, and a lot of harbor areas with industrial activity where there's PCB contamination. And that is what we'll be looking at here, but closer to home. This is in Baltimore Harbor. This is a study I did earlier on together with Kevin Sowers, who might be on the call, I'm not sure, where we looked at a core sediment that another colleague of us, Joel Baker, took from Baltimore Harbor at a specific location where he dated that core column. So we can see here the different slices of the core column on the x-axis have been dated, and you see the years listed there. And then we see the different homologs, groups of congeners, PCB congeners with different number of chlorines have been added up. And then on the y-axis is the total concentration. And if we look, we would have expected that this would be a curve that would have maxed out maybe around 1981, because that would be the result of the ban in the 1970s. But indeed, we see an increase towards higher and higher concentrations in the sediment in Baltimore Harbor. If we then move on a little bit in time and then look at that same sample, A is now our first column on this figure B. And then we have a number of graph samples from area bridges taken in 2006, so it's almost 20 years ago. We see an increase in concentrations beyond what we saw in the top layer of that column. So we can, and even if you pay attention to the y-axis, the numbers are completely different. So there's a lot more PCBs out in the environment now that early on in the 50s, when the main production of PCBs maxed out. So a conclusion from this study is that we know that there are current sources in this area here, Baltimore Harbor, but also in other places of the Chesapeake Bay. So it's not only a legacy PCB issue that we are dealing with. So the good news is that we are fortunate to live in a state where we are looking at these sources, and there have been TMDLs put in place for certain watersheds, and we'll get back to that later. So total maximum daily loads for PCBs. And that's exactly what you see on this map, some of the areas where there are PCB TMDLs in Maryland. And in this study that was funded by the Chesapeake Bay Trust, we looked in Anne Arundel County at different types of land use areas, because we were curious about the differences between the different types of development, not only time, but also types, and the relationship with the PCBs that we could find in those environments. So as you can see here, we're looking at 6 to 7 different types of land use categories, and we are classifying them as residential, commercial, institutions. They're also industrial areas. We are aware that industries have different signatures. So some of them have more light in kind of industry, and some are heavy. We also looked at energy sites and green spaces to have a diversity of the land uses where we could look into the impact. We also looked at what we call the land development era. So a number of these neighborhoods, for instance, are developed before the ban in 1970s, ban of PCBs. So we're looking at a number of these to see if there is a difference pre and post ban, but also the same with some of these other categories of land use. And you'll see a lot of that data very soon. So the objectives of the study was to assess the land use and time of development and the impact of that on the presence of PCBs in soils and stormwater sediments. We are interested in identifying potential sources, but then also to talk with practitioners and get information and ideas on what can be done to guide removal of PCBs in stormwater. Here you can see a number of the sites that we have visited and SUU has sampled from: residential neighborhoods, light commercial areas, and an energy site. For some of these categories, it was not possible for us to find a pre-1970s situation because there is an issue with redevelopment or change development. So that is something that we would also like to discuss when we move on. So back to where we are sampling. We are here, we are in Maryland, we are in Anne Arundel County, different locations there. You can see this map where we're looking at different categories that we just talked about. For many of these, we collected sediment and particles at the curb site or as close to that location that we are interested in without invading people's private spaces, of course, and so on. We also have to pay attention for having safe sampling practices and so on. And we had enormous help from Doug and Janice in Anne Arundel County. If you're on the call, thank you very much. And the sampling collection was largely driven by a discussion with people that are out there and know what's going on and have different suggestions to where we should look into the situation. So the number of collected samples that we're basing this study on are in total 45 that were collected over 2 years, 6 different land uses, 2 different time eras. And all of these were analyzed for a number of parameters, and they were analyzed in triplicate. So the data that you see are quite substantive compared to many other studies that we at least have found at this point. We have utilized the green spaces as kind of our control area, since there should not be so much impact from land use and time here. We are learning that that might not always be the case, but that is also something we can get back to. So just a single slide on the experimental process here. You can see to the left, Sue is out collecting samples. And then we bring them back to the laboratory in College Park at the university, go through a very thorough extraction of the PCBs where we keep track of what is lost and so on the whole way, so we can make sure that we account for everything in our data analysis. It is quite time consuming, but we want to make sure that we have a good quality QA/QC protocol in place. And if anybody's interested, we are happy to share. So the first part we moved into here was that for all of these PCB congeners, not all of them can be quantified and detected in each sample. And we have been involved in a discussion about what that actually means when we're determining the total PCB concentration, which impacts the TMDLs that we talked about, or the data feeding into the TMDLs. So what we're interested in was developing a method where lack of detection would not cause an underestimation or an overestimation. It has been common to substitute a number of half of the detection limit if there was an absence of a certain congener. However, that can lead to overestimation of the total PCB concentration that can have consequences further down. So part of what we're also looking into are methods that can help us get to a more accurate estimate of the actual data and the total PCB concentrations. And we've used a statistical method for that we are also happy to discuss. But in this, we have done work that shows what our detection limits are for each of these 209 different congeners, because they also vary. They're not equally easy to detect in this workflow that you showed before. And then we also know for this study what our method's detection limit is with the way that we have sampled. And all of this work is, again, to make sure that we are not overestimating or underestimating if we have zeros in our data. Okay, so if we look at the data now, so here is one of the plots that you'll see. We're looking at the different types of land uses. We're looking at the total PCB concentration. At the x-axis are the different types of land uses. When you see that it's pre-70s, it means that it's before the PCB ban, and then there's the concentration on the x or the y-axis. So what we can see is a quite a variation in the concentrations for many of these areas. So for particularly residential before the ban, there's a large variation, even more for industrial use areas before the ban. And you can see on the left the numbers here, the range of concentrations and the average. And you see the green spaces to the second from the right is quite low, but surprisingly not a whole lot lower than the institutions and commercial spaces after the ban. So we do have a certain background out in our environment in Anne Arundel County. So the summary for that is, if we're lining out the land uses, we have the highest concentrations in industrial, followed by residential and energy sites. The lowest are commercial and institutional, with green spaces being the lowest of them all. And we also acknowledge there is a large range of concentrations for these land uses. If we then take the same data and look at the development time pre and post PCB ban, we do see a clear signal that the concentrations have decreased in areas after the PCB ban. So that's good news, but we are still at quite high levels in some of these areas. So if we look at the reduction, for instance, in residential neighborhoods from pre and post, it's about 2/3 that it's gone down. Industrial pollution or PCBs from industrial areas have been cut in half, and then a little less for commercial and institutional areas. Some of these, the later one, the commercial and institutional, we know that it's a little difficult to determine the specific land use because there is redevelopment happening over time. And sometimes it's not easy to find the county records that show that. So we're quite happy that a lot of this have changed over time, that we do see a decrease. So then we get to sources. What do we know about sources? Well, we know that PCBs are in mixed aerochlors. That's largely the industrial applications, transformers, capacitors, and so on. But we also know that there's quite a few PCBs in building materials. We are also learning that there are other products, overall consumer products, that might have PCBs of some kind, which in itself is not assuring. And then we know also now that road paints contribute to a certain extent. We'll get into the road paints in a second. So this is a really messy graph, and we won't spend much time of it. The conclusion is we cannot see a clear signal from the aerochlors that have been used in industrial areas. They do not match up with our industrial, with our samples from industrial areas. So our PCB signal is not clear that it comes from industrial products. However, there's a large group, you see the non-aerochlors, the blue there, that must come from other sources, because that group of congeners do not come from the industrially used PCB congeners. So there's a lot we don't know, but there are things we do know, such as there are mixed sources, and there are some that we are not aware of out there that we have to go out and look for to try to look and limit the sources.
And can I interrupt you just for one moment? So we do have a question that came in, and I know you wanted to be able to address them as they came in. So we have a question that is, the detection limit is a wide range by about a factor of about 70. Is this wide range due to the different types of PCBs or different amount of like noise / background from the samples?
So the range is for, yes, as the question pointed out, these are for 209 different chemicals. So each chemical has their own detection limit, and the range is for all of them. that we show here. And it is indeed because the detection, the instruments respond differently to these 209 different congeners. So some congeners can be detected at very low concentrations because the chemistry turns out to be sensitive for the detector, whereas some of them, as also was pointed out, is just 70 times higher because of some factor of how the molecule looks. But that's a very good point. And that's, again, very nice work from Suyu that have looked at all of these detection limits to make sure that we have data that we can trust. But yeah, great question. Thank you. Okay, back to the sources. One of the studies that we are, the sources that we have been very interested in is road paint. And an earlier study have shown that we did before the pandemic, looked at yellow road paint. And we had 4 different samples of yellow road paint that shows and these were collected at the road, different concentrations. So the relative concentration varies from these different types, which could either be because there are different types of road paints or they have been sitting out in the environment leaching for different amounts of time. But nonetheless, we know from this study that the PCB11, that's a non-aerochlor, is a source in the environment. As you can see, it's commonly used at curbs and stripes in general, if you think about the traffic landscape. So then we looked at, well, if we know that PCB is 11 comes from road paint, yellow road paint, how much of the mass of PCB can we contribute to this source? And would that have any kind of impact on the overall mass that's running out into the environment? So you see some of the same data here. And as you can see in the green space to the left, we can't find anything. There's no trace of yellow or PCB11. And then there is a variation in these different areas. So that's the concentration on the left y-axis. If you go to the right y-axis, the percent there will show how many percent out of the total mass of PCBs in a sample can PCB11 contribute to. And that varies from almost nothing to about 1/3 of the total mass. We do not see any trend depending on the development time for the area, and there are very large variations. But that, I think, would be expected since we know that that this PCB11 comes from yellow road paint. So that is one thing that we can point to. Whether we will be able to do something about it is maybe something we can also discuss. So I'll just give a short introduction to a capstone project that some of our engineering students did, because the question for them was, can we calculate how much road paint there is in a watershed, and then use that to make a PCB calculator to estimate how much PCB actually comes from road paint in a certain watershed. They went in and did a lot of statistical analysis. They used different types of GIS and mapping, figured out the length of, here you see in the table, white and yellow road paint. There also happens to be PCBs in white, but that's a different PCB. And then they looked at the volume of road paint, and then they estimated the leaching coming from different types of road paints, and then used that to make a PCB runoff calculator that then, and here you see an example, can estimate the amount of PCBs coming from these 2 sources of road paint that runs into a certain watershed where the University of Maryland was in. And then you see on the x-axis the amount that we can estimate for 2024, one data source from another data source in 2025. And then the red bar in the middle is the TMDL that we have in the watershed that UMD is based in. So this is our first attempt to quantify at a larger scale how much impact PCBs and road paint have. And you can see it can be quite significant, which our data also showed. So at this point, this group of students graduated. We are continuing looking into data that can make this tool more sophisticated and hopefully also useful for practitioners. So then the other piece was, can we do something about the PCBs that are out there? And we have looked into bioretention cells, different kinds of treatment options, as you all are very well aware of. And the reason why we're also looking at that is because PCBs can be degraded by bacteria. They were naturally at one point, but now they've been produced in too high a mass for the natural microbes to keep up with them. But there are two different zones that we're looking at. The top of soil is generally aerobic, so there's air and oxygen available, and the bottom part is anaerobic. So if we look at this and then connect that to PCB microbiology, and this is a busy slide, I apologize in advance. The two important things that we need to look at is, depending on the type of chemistry that our congeners have, if there are more than 4 chlorines on the molecule, then we need anaerobic conditions for biodegradations, whereas if there are 4 or less chlorines, then we can use aerobic degradation, which is a lot faster. So we need both processes to end up at complete mineralization that will also remove PCB mass. So that is what we're looking at. We're trying to get rid of PCB mass. And then if we go back to our data set, we're looking at the same kind of samples here. Is there any sign of PCB degradation? And here we're looking at whether there are samples that have more than 4 chlorines and samples lower than 4, because the ones that have lower than 4 are readily are able to be biodegraded right away with bacteria that are already out in the environment. So if we can identify places where these things happen, then we can maybe help the process along with some of these BMPs when we look at that. So for a third of the samples that were collected, it would be possible for us to push aerobic biodegradation, whereas the other ones will need to go through both of these processes. So there is hope out there. We had a discussion with Doc last week, and he asked me to also talk a little bit about PCB toxicity, because when we're talking about mass, we're talking about mass from all 209 PCB congeners, but it's only 12, if we can say only about that, that are particularly toxic, that's causing the majority of the issues in the environment. And these 12 we call dioxin-like because of their chemical structure. Depending on the environment they're in, they have different impact, which we call toxicity equivalency factors. And it's different whether it's a mammal, a fish, or a bird that will be impacted. So we also looked at something that's called the safe sediment value. And in this case, it can be determined to be 20 picogram per gram of sample. We have a source for that. And then we looked at our sample here to see how many of our samples would be above that safe sediment value. And it actually turns out that there are only 3 out of 45 samples that would exceed that toxicity limit. That's still bad. They all happen to be the older development situation, so residential and 2 industrial. So residential would, in my mind, be a little bit of a surprise, maybe not so much for industrial areas. But the take home is that not all PCBs cause toxicity. And now we have a good idea at where we should look to figure out the impact on the environment, and that is in the older development areas. And I think I might just give an introduction to this slide.
Yeah, we have about 10 minutes left.
Okay.
Okay. I just want to give you a heads up.
I will make this short because this is from another study that we have worked on where we're trying to look at all the different options we have to remove contaminants when we're talking about stormwater, straight from source reduction, pre-treatment, traditional bioretention cells, but also we looked at polishing treatment in the laboratory. And a lot of what we have started to think about is where can we take the most mass out? Where does it make the most sense to put our effort to reduce the mass of contaminant? And in this case, it's PCBs. And one of the things we have looked at is street sweeping. And the data here is not from this study, but there is quite a lot of data showing that there's large removal efficiencies if we look at a treatment train approach, but also if we look at source reduction in itself. And this is a back of the envelope calculation that were done together with Anne Arundel County this past week, where they looked into how much debris is collected with street sweeping. We have several assumptions about dry matter and the different concentrations of PCBs in the material that's swept up. Here we used our data and assumed 50 nanograms per gram, which is actually low to mid range of the concentrations we have found. So by looking at what the data that's available, we can calculate the amount of PCBs that street sweeping is taking out, which is not to be underestimated. And then can be a discussion about where should that street sweeping then take place if we want to have the largest impact for the effort that we're putting in. So that is something I think would be a good discussion later on here. So then just to sum up, in this study, we looked at a number of different locations. We can see that the post-1970s development areas have lower PCBs. We know that up to a third of a PCB mass in certain samples come from PCB 11. We also know that there's, in a few samples, toxicity that was exceeded. We know that stormwater carries PCBs. So a lot of what we would like to also discuss with you, what can we do? Which tools do we have to reduce? And then the next steps that we're looking at, is there an impact of soil and particle types out there? What is their contribution from other road paint colors? Because we have more than white and yellow around. Literature points to those two colors being the dominant, but which other sources are there? Because no matter what we do, there is more than half that's not to be contributed to road paints. So we have to look into other sources. And then when we're thinking solutions, we cannot forget other contaminants that also must be handled. So is there a way to think in nutrient removal with these biological processes that I showed for the PCBs so we can design smarter BMPs that can handle more contaminants than what we're thinking about right now? So a lot of students, a lot of people have been involved, organizations. Thanks to everybody for contributions before, during, and now we're getting into even more discussions. And also the capstone group that gave the first prototype, if we can call it that, of a PCB calculator. And just making sure that all of you know that we appreciate if you have ideas that can be used for capstone projects of any kinds, just please reach out. Our students are so interested in dealing with real world issues. Thank you very much. I hope we can have a good discussion.
Yeah, Thank you so much, Dr. Kellerup. That was fantastic. We do have a question right now. I didn't want to interrupt you as you were going through, but Sabrina has another really great question. How is the leach rate estimated for the PCBs in road paint?
That is a great question. So these students used the literature there was, which is limited, I will say that, and then estimated how much that would, how long that would take. And then they talked to their traffic colleagues and looked into the guidelines for how often road paint must be replaced, because it seems it doesn't sit in, at least in all places, does not sit there forever. So unfortunately, it seems that we are reapplying a lot of fresh paint with a lot of new PCBs quite frequently. So we never get to, zero leaching. So there are some estimations and calculations in there and assumptions that would be good to discuss.
Excellent, thank you. We have another question from Emily. She said, great presentation, thank you. Can you describe how you selected your sample sites in the different LU categories? And also, were they randomly selected or targeted? And did you look at the drainage areas of the locations were they within the same watershed?
Excellent questions. And here, the real experts here are Doug and Janice from Anne Arundel County, because we worked with them quite a bit to get a representation of sites, some that were targeted, but also some that were more representative. And they're not all in the same watersheds. There are some that they had a specific some locations in certain watersheds that they had specific interest in, and some that were more general. So we try to cover all bases, more or less. So I wouldn't say that these are all areas of high concern. We have both. So we would, because that, would cause a bias. We would probably overestimate if we went for locations of interest only, but we didn't.
Excellent. Thank you. And Sabrina has another question. Why is there still PCBs in road paint if PCBs were banned in 79?
That is a super good question that I have asked myself many times. It so happens that that ban in the 1970s were for PCBs to be the main product. So if you were set out to make transformer oils that would have PCBs as the main chemical, that was banned. We have now learned that a lot of the products that are chemically synthesized, and PCBs are a byproduct, such as the synthesis of yellow pigment for road paint, then PCBs can be present, and I assume are allowed. Why? I don't know. And more disturbingly, or equally disturbingly, is that that yellow pigment is also allowed in pigment for brochures or paper cardboard, and if we're really unlucky, in certain type of food products. So we should really ask ourselves why we still have PCBs present.
Wow, that is incredible. I had a question while we're waiting for other questions in the Q&A box. Again, if anybody has any questions, go ahead and drop them in the Q&A. I had a question about at the beginning, you were talking about the study that involved collecting sediment in the harbor itself and seeing that, the PCBs should have, at least leveled off or capped. So those sediments, were they collected like on roadways around, or were they actually collected from the bottom of the water?
They were the top layer. They were collected with a Poner sampler thrown in from a bridge. So those were the actuals. That's what the fish will experience at those locations in the bay.
Okay, great. And was there any question about, because the harbor is dredged regularly, was there any question about how that could have affected the layers, or anything like that?
Yes, and that's an excellent question. And we know that it's not only dredging, it's also resuspension from the big cruise liners and whichever ships happen to come to Baltimore that can impact that. So those locations that you saw on the graph, they were at tributaries because otherwise we couldn't get access. So I don't think there would be any contamination that could be attributed to that. But for sure, out in the main water body, there is a lot of replacement or replaced PCBs available. And one of the big questions I have about this is also, what does the Bay Bridge decommissioning and reconstruction do to that? And I'm not sure that it's on the forefront of people's minds because of a whole bunch of other issues, but that will impact what we see and what will be resuspended in a water column for many years.
We have another question that this is from Skylar. What can be done at the individual level to help combat and spread greater awareness of PCB impact?
That's also a great question. And I don't think we, with the data that we have here, can talk a lot about that because we don't know what the other sources are that we see. We know that some are coming from industrial PCBs, some are coming from road paint PCBs, but there's still a large chunk that we don't know exactly where that's coming from. And that could largely be from building materials. But it could also be from atmospheric deposition that we don't have, as an individual, we can't do much about that. So a lot of it is to get to know more about what's in building materials. We are very good at trying to recycle and reuse building materials. Maybe we have to be, I wouldn't say careful, we have to be aware that in that way we might potentially spread sources when we are trying to reuse some of our building materials. There are some studies in Europe that have shown that crushed concrete can contain certain amounts of PCBs, but then you can also discuss whether they are then kept. If they're used for making a new road, how many years are they then going to be kept there, right? So we do need a whole lot more knowledge about this. And that's also for what we can do as individuals. Be aware about the products that we purchase and consume and how we behave.
Excellent. That's really, really great advice. And I think it is very interesting how you're saying sort of that legacy of materials that we once used for building, they're still on many of these buildings, right? And are they still leaching? Are they still contributing on their own, even though we're not using it in, modern building materials? It's an excellent question. All right, well.
So we do know that there is a number of PCBs in schools, for instance, which is why we have the institutions. Is a lot of that is hospitals and schools, because we know that they are, unfortunately, at least in our state, a lot of the schools are from way before the ban. So that's a specific category of interest.
Wow. That's also frightening that it's in our schools, Well, I just want to say thank you so much. Dr. Keller, this is really, really amazing, fascinating work that you're doing. And I love that you're very open to hearing, from different aspects of stormwater and seeing how people can contribute and ask more questions to be able to further this research. I think that's amazing. If anybody does have questions that they want answered, you think of it later on, you could always use the email that's in the chat and email me and I can get that to Dr. Kellerup. She also has her contact right on here. You can email her directly as well. She's more than happy to discuss things and hear from you as well. So thank you again. I hope everybody enjoyed the webinar, and we look forward to having you again for our next webinar coming up on July 8th as well. So I'm going to stop our recording.